we have from Martina Crispo. She is responsible for the laboratory where they produce and modify mammalian embryos of different species. And she's working at the Pasteur Institute in Montevideo, Uruguay. And we're glad to have you here, Martina. So please, you can start sharing your presentation. Thank you. You have 30 minutes, out of which 20 for the presentation, and the rest will be for the discussion. Thank you, Martina.
Okay, thank you. I will try to share the screen now. I think now I'm sharing, and thank you for inviting me to give this keynote lecture at the ARRIGE annual meeting. I hope you hear me well. And yes. I tried to make a simple presentation of all the incredible advances we have heard over the last year on gene editing field. So let me say that it was a huge amount of work, because there are many great papers in this last year, and I will try to explain in simple words this, and at the last minute we can discuss and we can exchange further for the ethical point of view. So let me switch here. Okay, let's see if it works. Yes. And as Luis said in the General Assembly, I cannot start this presentation without acknowledging Jennifer Doudna and Emmanuel Charpentier for awarding this year the 2020 Chemistry Nobel Prize for the development of a method for genome editing. And this was great news for our field. We were all very happy, although there were some researchers that were leave out of this prize, but I think that always happens in the Nobel Prize, so we are very happy to receive this prize for our field. And this is just a reminder of how CRISPR works, and this is the basic work of CRISPR, because now it has changed a lot. But basically, the classical approach of CRISPR is the break in the double-strand DNA. And this break can be repaired by endogenous repair mechanism of the cell, by non-homologous enjoining, creating a knockout, or by homologous direct repair, creating a knock-in. And also, CRISPR can mutate RNA, not only DNA. So this was described a few years ago, and is the same approach. But CRISPR, you only need to add some PAMRs, and CRISPR can also mutate single-strand RNAs in the same way. And this is coming more and more complicated nowadays, because the structure of CRISPR is changing, and they are discovering new CRISPR systems. So I'm going to explain in this lecture about this. What are the applications of gene editing? There are several applications. One of the most widely spread applications is the generation of animal models. In this case, I work with the generation of animal models, mostly mice, but also large animal models. There are hundreds of papers coming out using this technology to generate, for example, cancer or neurodegenerative disease animal models, models of infertility, several models of human or animal disease, also models for enhance animal productivity when you work with livestock species. And let me show you our last large animal model that was published this year in Scientific Report. This is a sheep we have generated in collaboration with a group here in Uruguay and in France, in which we mutate with gene editing the autoferlin gene. The autoferlin gene is a gene that appears mutated in humans and impairs the audition of humans. So we have generated this large animal model to test different therapies for recover audition in humans. So for that, we have transferred around 1,300 embryos, and we have obtained good editing rates. Around 18% of the lambs born were edited. And from this 18%, we have obtained around 62% of lambs with the mutation we wanted. And so these chips are available to test different therapies against deafness. What else? What can we do with CRISPR? We can use them for human immunotherapies. This is a field that is growing a lot in the last year. In this case, for example, this is an article that has recently appeared from Liu et al. And they edited T cells, normal T cells from patients with refractory non-small cell lung cancer. In this case, they retrieved the cells from the patients, and with using the classical approach of CRISPR ex vivo, they electroporate the Cas9 and the single guide RNA to mutate the PD-1 protein. PD-1 protein, when you mutate this protein, you increase the immune reaction. The PD-1 is an immune checkpoint. So they put again the cells in the patient. This is a clinical trial in 12 patients that receive again the transfusion of these mutated cells, and they were monitored for up to 96 weeks, and they found no severe treatment-related adverse effects in this case, and very low off-target editing events. And that accounts and start to add more information for the immunotherapies using CRISPR. Another approach that has recently came this year using the same approach with CRISPR. In this case, this group edits also ex vivo stem cells from a patient with HIV, and they mutate the CCR5 receptor, and they put the cells back in the patient. There was only a single patient. In this case, in this other paper, they used three patients with refractory cancer, and they use this therapy as a composite therapy, and they do the same approach. They retrieve the T cells and they mutate the T cells ex vivo for the immune checkpoint also, and they put the cells back. And they found in all these papers that the efficiency varies significantly across target genes and patients. It depends on the gene where you want to mutate, is the efficiency you find. In these two papers, for example, the mutation rate was about from 5 to 20% of the cells had the edition they wanted. And there is a need to evaluate the long-term safety of CRISPR. Of course, these are very short reports, clinical trials. They found that this technology is rather inefficient, and they say they are ready to test base editing and prime editing. So we already heard about base editing some years ago from some groups working in this, where you can specifically mutate one single base pair in the DNA. And now lately we have heard about prime editing. So what is prime editing? Prime editing is a very sophisticated tool that has recently appeared from the group of David Liu. This was published in October last year. And in this case, they have managed to use an impaired Cas9 fused to a reverse transcriptase, okay, and a prime editing guide RNA called pegRNA. Here you can see the tool, okay, with the Cas9 fused to the reverse transcriptase and the pegRNA. And with this very, very precise tool, they can manage to do targeted insertion, deletion, and they can arrange 12 types of base substitution without making double-strand breaks in the DNA or without introducing donor DNA templates. So this is something that is improving a lot the field of genome editing. And they found higher or similar efficiency and fewer byproducts than homologous direct repair, and complementary strengths and weakness compared to base editing. So, and they didn't find many off-target editing places, much lower off-target than Cas9 nuclease. With this, they proclaimed that it's possible to correct up to 89% of known disease-causing gene mutations that are due to a single base pair mutation. So, with prime editing this year also, recently in October, this group has managed to mutate cells. They obtain cells from patients with several diseases like Wilson disease or patients that had deficiency in the DGAT1 gene, and in this case, they obtain the cells and prepare in vitro organoid culture models, and in this case, they recover the disease-causing mutation in these intestinal or liver organoids using the prime editing with greater precision than homologous direct repair. So they also compare in the same paper base editing against prime editing, and they find that base editing is more reliable than prime editing. That means there is a lot of work to do also with prime editing because it's very recent, this technology, and they didn't find any off-target effects. So this is something new, this is working. We have to wait for more information regarding this prime editing tool, but it seems that it's getting more and more, the gene editing field is getting more and more complicated. So we have base editing, we have prime editing, and we also have dual base editing. What is dual base editing? Based on the prime editing, on the base editing that was originally published a few years ago, now some researchers working in plants, maybe we will talk about that later, managed to fuse these 2 deaminase with Cas9 nickase and make the 2 editing in the same target site. That means they convert CT and AG on the same target time, not only one editing. And they found less adenine activity, more cytidine activity, and less off-target effect than base editing. And lately, in June, there were several reports in the same number of Nature Biotechnology, like 3 reports using this dual base editor that seems to work also in human cells. So we can mutate DNA, we can mutate RNA, and we can also mutate mitochondrial RNA. So you can see this is getting more and more complicated and sophisticated. This year, in May this year also, the group of David Liu, he's a very active researcher in this field, has published the possibility to mutate mitochondrial RNA using base editing. In this case, these researchers use a toxin from a bacteria called DDDA. This toxin, they split the toxin in half, okay? So in order that not let them be toxic, and they fuse it to a TALEN and an uracil glycosylase inhibitor. So in this case, they managed to assemble this RNA-free DDDA derived cytosine-based editors. Okay, and with these base editors, you can catalyze CG to TA in the mitochondrial RNA. And they found that this high target specificity and precise mitochondrial RNA manipulation. In cells, they found that this makes some changes in respiratory rates and oxidative phosphorylation. And this can have broad implications to study and potentially treat mitochondrial disorders that you know are some very complicated disease to treat, because until now there were only possibility to destroy the whole mitochondria with the consequence that can have. So now we have this new tool that can aid to avoid these mitochondrial disorders. And again, for editing RNA, another system that has recently published uses another approach called ADAR. ADAR accounts for adenosine deaminase acting on RNA. And with this new tool, those are human proteins that can mutate double-strand RNA. With this new tool, we can change in the double strand adenosine for inosine. Or if you use this other approach, ApoVC, we can mutate C. We can mutate the C also. So these proteins does not generate, at least they said, no immune reaction in the patient because they come from human. They are human proteins. And with this, they proclaim that they can correct human diseases like muscular dystrophy, cancer, acute pain, cholesterol levels at the RNA level with high efficiency and high specificity. They also state in the paper that the regulation of this therapy should be easier because there are many RNA or already approved drugs in the market. So as you are going to mutate only RNA, the regulation should be easier. And amazingly, another discovery from this year from the group of Jennifer Doudna is a new CRISPR system found in huge phage. Phage are virus that infect bacteria, and these huge phage from around 500 kilobase have another system of CRISPR system called CRISPR-Phi. Let's call it CRISPR-Phi. And they found that it is a hyper-compact genome editor. You can see it here in the upper. I hope you can see the mouse. You can see it. It's very small. It doesn't have the Cas protein genes, and it's half the size of, for example, Cas9 or Cas13. So it's very small. It accounts for a 33% of cell addition in this paper. And we still need a lot of information about that because we don't know if it's more specific than previous CRISPR-Cas system. It's better for in vivo therapy, maybe yes, because it's smaller and we can deliver it easier to the patient. And finally, we have to take into account CRISPR for this new pandemic, COVID-19, that we are all aware of. Does CRISPR work for COVID-19? I have to say yes. I have been reading lately a lot of papers working against COVID-19. In this case, there are several approaches that have been used before for DNA or RNA virus, like Sherlock from the group of Fen Sang, or Detector from the group of Jennifer Doudna. In this case, they use Cas13a or Cas12 for another virus like MPV or dengue or Zika. This method seems to be rapid, inexpensive, simple, and one of the advantages of this method is that no equipment is needed, no expensive equipment is needed. So the idea is to have this method anywhere of the world, and that anybody can do it. So how do they work? I will try to explain the rationale on this method. In this case, we have the Cas, for example, for the method of Sherlock, we have the Cas13a enzyme linked to the spacer and the target, and this enzyme will cut the viral RNA in a specific precise site, but after it cuts the RNA from the virus, it will recognize several similar sequences around, and it will very, very precisely and efficiently start to cut all the similar RNAs. And in this case, they add a fluorescent sensor, RNA sensor, linked to a repressor in the same system. So the enzyme will cut the RNA from the virus and will cut also this RNA that is similar to the virus and will release this fluorescent dye, and this will, by colorimetry, we can detect then the RNA from the virus. And detector is from the group of Doudna, is very similar. it has only a previous step that we have to take into account because it uses Cas12 and Cas12 cuts DNA. So in this case, we have to convert RNA with a reverse transcriptase to RNA and then to a DNA, sorry, and then it works in the same way with this fluorescence sensor. In May this year, the FDA approves a rapid COVID-19 test that uses CRISPR, the test from Fensang, as an emergency test, and this has been called Stop COVID, and it's very simple to use. You have to take the swab, the sample, or the saliva sample, and then you put the sample into the Sherlock system, and the only equipment you need is this heat Sherlock that will heat for 60 minutes at 60 degrees, and then you just dip a strip into the system, and you can check if you have one or two guides here. If you have two, you will see that it's positive. If you have one, you will see that it's negative. And lately, we have detected that we have heard about an approach from Jennifer Doudna that accounts for a new test for coronavirus that can detect coronavirus in just 5 minutes. This is very new, and this uses Cas13a using a mobile phone, and we will see if it works, but it will be great if it works. So for treatment, CRISPR can also work for treatment. In this case, there is a system called Pacman, and Pacman can detect RNA from the virus very efficiently using CRISPR-based strategy. They will produce inhibition and degradation using Cas13d. There are a lot of acronyms for this system. Pacman is one of them, and they tested in vitro with a virus, and it was very, very efficient also. With 6 CRISPR RNAs, you can target more than 90% of all coronavirus. But we still have ethical concerns. This is very recently reported. I'll look at the date, October 31, because there were some researchers that report some on-target edits, large-scale and intended DNA deletions and rearrangements in the area surrounding the targeted sequence in human embryos. So we need a lot of information regarding these technologies, because we can find also these on-target edits that can be very, very detrimental. As concluding remarks, let me tell you that this gene editing system has versatility. There is a new therapeutic paradigm targeting diseases at the RNA level. We have to take into account delivery, administration, safety, preclinical study design, drug manufacturing, and economic and regulatory issues. But we still have a long road ahead. So with that, let me say gracias, and I'm ready to discuss with you further and exchange the ideas about these novel technologies. Thank you very much.
Thank you very much, Martina. You can stop sharing your presentation, and so we can, because there's a couple of questions already arrived here. So without further delay, I will ask you the questions. So Guillaume Levrier is asking, why did you choose to work with lambs to do animal models? Is it easier technically to do than macaques?
Yes, I think yes. We have any facility here for macaques. It's nice, but we have a strong collaboration with a group of researchers here that work with animal reproduction, mostly in sheep, and that's why we do this collaboration with them, and so we produce with them the embryos in vitro, and we mutate them. the system is really similar than mice, and then it's more complicated, the embryo transfer and all the things that this group does outside.
Thank you. Then there is another question from Guillaume, says basically it is due to institutional processes, but I guess you already answered. You already answered this. Then we have Shalini Reddy, and she's asking regarding prime editing. Won't the reverse transcriptase in prime editing cause non-specific activity?
Well, I don't know, because this is very, very new, and the idea is that it's very specific of the target site you want to edit. I hope it does not make any other edit, but we still, as I said before, we still need more information regarding these new technologies, and maybe somebody there can ask more properly that is using prime editing. But as I said, it's very new.
There are few cases in which prime editing has been used outside the David Liu's lab, so we need to pay attention.
Mm-hmm.
So there is another more specific question from Shalini Reddy on prime editing, but then we can skip this and we can go to Guillaume Lebrier. He has another question regarding the diagnostic tools, and he's asking whether any of these Cas-based diagnostic kits are available in the market.
The only one I know is the one from Feng-Sang that is called for COVID. You are asking, right?
Yes.
This is in a small amount of patients, and this is, but it's not ready to be used for everybody. And it seems to work.
They seem to have an FDA emergency approval, but nothing. It's at least in Europe, no one of these different kits has been authorized.
No, we have a LAMP approach here that can give you results in 1 hour, but also it's not for everybody. So they managed to have it for only a few cases.
Okay, then since we don't have more questions, we can go back to the question I skipped. So that's Shalini Reddy again, and he's asking regarding prime editing whether because of the long RNA is also binding to the protospacer, whether this might have some implications in reduced efficiency.
Sorry, can you repeat the question,
yes,
Luis, because I didn't understand if the long RNA.
No, because we're having some difficulties with the connection, Martina, and but I'm repeating. So basically, for the prime editing, what she's asking whether because this long RNA is binding to the protospacer, whether this might have some implications in reducing the efficiency.
I really don't know about that, but maybe yes, can have reduced the efficiency, although I think that there are still a lot of field to work in this prime editing new technology to improve the efficiency, because they say base editing is more efficient than prime editing, but maybe yes, you're right.
I think I have a question. What you have presented, and I do entirely agree, it looks like the base editors are the next big thing. So base editors in all the different varieties will probably be replacing most of the standard uses with traditional CRISPR-Cas, because the efficiency is being increased and the specificity is also being increased. Do you think it's the time for base editors? Martina.
Sorry, the connection is very bad and I'm having problems with that. But I think, yes, base editing will replace the standard approach. We would like to try it very soon in our animal models, because they seem to be much more efficient. And as I see during these 2 weeks that I have been reading information, it's getting more and more sophisticated and more efficient. So it will be maybe the future for that.
Thank you. So there is yet another question from Prime Editing, a lot of interest in Prime Editing, and they say whether this has been.
I'm not an expert in Prime Editing, but
no, yes,
I read and there's not many information.
No, let's discuss whatever the discussion. The question is regarding whether Prime Editing has been used on human embryos.
Not that I know, and maybe we will have more information soon. But the only reports I found was that the first one and this one in human organoids from liver and from intestine, but not in human embryos.
Thank you. Well, we will have to finish. So muchas gracias, Martina. Thank you very much for your nice presentation. I think it was a good start for this meeting. So now let's move now to the next